everyone. My name is Ankur and welcome to RJPT Simlab simulation. In this video, we will study about the bioassay of histamine using Guinea Pig Ileum by matching method. Now, let's start with the video. But at first, let us see the description of this experiment. The bioassay of histamine using Guinea Pig Ileum is a pharmacological method to evaluate histamine's biological activity. The Guinea Pig Ileum, a smooth muscle response to histamine by contraction, which is used as an indicator of its potency. The above statement says that the bioassay is a pharmacological method which is used to evaluate histamine's biological activity and the guinea pig ileum, the tissue we are using, is a smooth muscle which responds to the histamine by showing contraction as an indicator of its potency of the histamine. And in this bioassay, tissue preparation from the ileum are exposed to various concentrations of histamine and the degree of contraction is measured. The method helps in determining the concentration response relationship and the potency of histamine or histamine antagonist. It is commonly used for evaluating histamine's role in allergic reactions or testing the efficacy of antihistamine drugs. The above statement says that in this bioassay, we will do the tissue preparation of the ileum and the tissue is exposed to various and different concentrations of histamine and the degree of contraction is measured according to the concentration of the histamine. Then this method helps in determining the concentration response relationship and the potency of histamine or histamine's antagonist drugs. And it is a very commonly used method for evaluating histamine roles in allergic reactions or efficacy of the antihistaminic drugs. Now let's see the equipments required for this experiment. At first, we have our animal guinea pig. Second, we are using the drug histamine stock solution of 10 microgram per milliliter. Third, the instruments we are using are student organ bath and chymograph. And at fourth, the physiological salt solution, which is our thyroid solution. We are using the thyroid solution so that our tissue is kept preserved and is kept away from degradation during the experiment. Now let's see the principle of this experiment. The principle of bioassay of histamine using the guinea pig ileum is based on the contraction of smooth muscle tissue in response to histamine, which means contraction of the smooth muscles in response to the histamine's different concentrations. And when histamine is introduced to the isolated guinea pig ileum, it binds to the histamine receptor, the H1 receptor present on the smooth muscles, causing a series of biochemical events that leads to muscle contraction. The histamine acts and muscle contraction takes place, and the degree of contraction is proportional to the concentration of histamine. The degree of contraction, the amount of contraction takes place is directly proportional to the amount of histamine, the concentration of histamine. We are using different concentrations of histamine. By comparing the response of the ileum to known concentrations of histamine, the potency of a substance being tested can be determined. This method is used to assess the physiological effect of histamine and evaluate the efficacy of antihistamines. This is the observation table of this experiment, which shows the concentration of histamine, that amount added of histamine in student organ bath, and the concentration of histamine in the student organ bath with the solution filled in the student organ bath. Then the response, which is recorded from the movement of the frontal lever upward and downward on the chymograph in millimeter, and then the response percentage of the response. Now let's see the procedure. These are the steps according to which we'll perform our experiment. We will discuss these steps while performing the experiment. Here we have 20 steps to perform. This is the calculation part of the experiment. We have the formula A1 into X equals to A2 into Y, where A1 is the concentration of test, X is the dose of the test selected, A2 is the concentration of standard, Y is the dose of standard that matches with the response height of the dose of the test solution. So the shifting goes and A1 equals to A2 into Y upon X. And after recording the values, we will put the value and we will do the calculation and we will find the unknown concentration of the test solution of the histamine. Now, the conclusion part. The bioassay of histamine using guinea pig ileum is a reliable and effective method to study the pharmacological effects of histamine on smooth muscle contraction. It provides valuable data on histamine's potency and allows for evaluation of antihistamines or other compounds that may modulate histamine activity. The contraction response of the ileum serves as an indicator of histamine's biological effects, and through careful analysis of the concentration response curve, researchers can quantify histamine's activity and assess the therapeutic potential of histamine related drugs. The above paragraph says that this bioassay is a reliable and effective method for the study of pharmacological effect of the histamine on smooth muscle contraction, and it provides valuable data. And we can find the potency of histamine of the unknown concentration and can modulate histamine activity. And the concentration response serves as an indicator of histamine's biological effect. And through the analysis of the concentration response curve, we can quantify the histamine's activity and assess the therapeutic potential of histamine related drugs. Now, let's perform our experiment. For that, we will perform this experiment in our RJPT Simlab simulatory laboratory. As you can see, this is our RJPT Simlabs lab. From the movement of the arrow keys, we can move throughout the laboratory. And now let's discuss our instruments, which we will use during the experiment. As you can see, at first, we have our student organ bath. Our student organ bath is the place where we will keep our guinea pig's ileum. It is connected to a reservoir which supplies the thyroid solution so that our tissue is kept preserved. This is the frontal liver which is connected to the student organ bath, which over which we will hang our tissue so that it moves upward and downward according to the contraction of the smooth muscles of the ileum. This is the aerator part. The aerator is connected to the student organ bath so that it can supply the continuous supply of oxygen and carbon dioxide to the tissue so that it is kept away from degradation and it is kept preserved while performing the experiment. We have a chymograph drum. The chymograph drum is connected to a Sherrington revolving drum. The Sherrington revolving drum is a device which is used for the recording physiological signals and it rotates and controls the speed, allowing accurate time-based recordings. It rotates the chymograph drum and over which our graph is drawn with the movement of the isotonic frontal lever. The frontal lever is attached to a casual weighing balance, so that the movement of the frontal lever is prepared upward and downward. And while we hang our tissue to the frontal lever, the balance mediates and equilibrates the weight, so that it does not hang up while hanging the tissue. Now let's move forward. Here we have a petri dish which also contains the thyroid solution over which we will place our isolated guinea pig's ileum. It is also connected to the aerator so that a continuous supply of oxygen also supplied to the petri dish while isolating so that it does not degrade during the experiment. We have to perform very carefully so that it does not contaminate or degrades. Here we have our dissecting tray over which we'll place our guinea pig so that from where we can isolate our guinea pig ileum. We have our test tube holder which contains a test tube which contains a histamine stock solution. We have our scissors, pithing rod, forceps, suture scissors. With the help of these, we will cut open our guinea pig and isolate its ileum. We have digital pipette. The digital pipette is also known as micropipette, and with the help of this micropipette, we will put various concentrations of the histamine into the guinea pig ileum for the recording of the response of the histamines on the smooth muscles.